It's a great pleasure and privilege to introduce Allan Doctor, who is here today from St. Louis, where he's the Chief of Pediatric Critical Care. Allan has a very distinguished background, having trained at the University of Virginia and then at Boston Children's Hospital, both in emergency medicine and in critical care. He's done outstanding research in neutrophils with John Arnold at Boston Children's, and then in red cells switched from the white cell to the red cell to a large extent when he moved to Charlottesville to be a junior faculty member. He's been extremely successful in his work in oxygen delivery, red cell biology, and the modifications of red cell and hemoglobin bioactivity by nitric oxide, oxygen, and other ligands. And he was recruited to be the chief of pediatric critical care at WashU by Alan Schwartz, and he has really thrived there. And it's a real pleasure to see the quality and quantity of his publications, 37 publications now, publications in PNAS, JCI, New England Journal, very accomplished. The accomplishment of his that he should be proudest of is that he now teaches a 400-level chemistry course at Washington University, and hopefully he'll provide us some information about what he teaches there. He has numerous NIH grants on which he is the co-I, and Children's Discovery Institute from WashU, which is the PI. He also has a number of interesting industry grants, and I'll just read some of the companies: Terumo, Integrion, and Asclepion. So I want you guys to ask him in the question and answer session about those industry grants and what he's discovered. It's kind of interesting. It's a great honor also to have him as part of our program project grant with the Titus Group, and he's here today partly in that capacity, but mostly to provide some new data, and he says controversial data, about red cell biochemistry. And we are no strangers to controversy, and we're excited to see it. So Allan?
Thanks, Ben. It's actually quite an honor to be here and be part of this program. In fact, quite a bit of the foundation work for the data that I'm going to show you was done by Ben and people in this program. So it's quite, in fact, fun to come back and show sort of what we've been able to do with this. So what I want to talk to you principally about today is energy metabolism in red blood cells, and how the energy metabolism in red blood cells is in fact dynamically responsive, and I think possibly controlled by S-nitrosylation. The branding and disclosures for you all. And I want to begin by asking you to forget some prejudice and preconceptions about red blood cells, which are sort of a forgotten and misunderstood cell type. And so quite often people think of red blood cells as just simply cells that are relatively simple, that subserve cellular respiration by simple gas transport alone, and are not themselves involved in vasoactive signaling, nor do they really coordinate physiologic responses to stress, but really just bystander tissue that perform a sort of pedestrian function. In fact, people think of it as sort of a static tissue without much adaptive capacity in their function and metabolism, and that they are relatively uniform, meaning one red cell is more or less like another red cell. I mean, they all more or less look alike, and that there isn't much diversity within the population of red cells, either in how they function or in their specific role. Moreover, the conventional disease model is not really thought to apply to red blood cells. In fact, nobody really thinks too much of acquired disorders of red cell function. Really, their principal hemoglobinopathies or genetic and inherited disorders of red cell structure and function. And instead, I'd ask you to consider the following propositions, really to try thinking about red blood cells as an organ, which other people have named an erythron, which really has a very important role in coordinating physiologic responses in the whole organism and at the small tissue level to achieve O2 delivery homeostasis despite significant variation in either O2 availability, meaning in lung disease or in hypoxia or altitude, or on demand with physiologic stress or illness. And this happens at several levels, as I mentioned, at the organism level, where red cells have an adaptive response and can vary O2 affinity and or deformability and rheology. At the organ system level, as many here who have shown that red blood cells can coordinate variation in respiratory drive, in baroreceptor response, and myocardial performance in accordance to need to either improve O2 uptake in the lung or delivery to tissue. And at the tissue level, red blood cells can match regional flow to need at a small local level by controlling the NO bioavailability in plasma. Also, they influence genetic responses in tissue, as we've learned that red cell-based signaling is involved in HIF stabilization and then influencing our program response to hypoxia. And that these responses are robust across multiple timescales, both in the developmental level, and it's adaptive to changes in baseline in human physiology over the course of weeks or months, and also very acute changes in terms of physiologic stress on the millisecond and second level. And that this is also versatile in that it affords concurrent and spatially distinct responses, meaning that some red cells can act one way in the lung and another way in the liver, et cetera. Likewise, as I mentioned, many people think of the conventional disease model not applying to red cells, suggests that perhaps red cells do exhibit acquired dysfunction, as do other organs. And many examples are published: sepsis, sickle cell disease, storage, and I'll show you some of those data. And that at the root of this problem are often we find oxidative modification to key regulatory and structural proteins. And at the root of the oxidative modification really is energy failure in depowered antioxidant systems. And that really is going to be the focus of the information that I'm sharing with you today. And lastly, I'm going to suggest that really there is adaptive control of energy metabolism in red cells, and that this is required, in fact, to afford increased O2 delivery, the physiologic responses that I mentioned up above when there's physiologic stress. And that likewise, the erythron, in fact, has subpopulations with different function, and that these also afford this versatility as our body experiences changes in demand. So overview for the information that I'm going to share. Briefly touch on red cell control of NO bioavailability in plasma. That's obviously something reasonably well discussed here. In that NO is trapped and dispensed by red cells in response to physiologic oxygen gradients during circulatory transit, and that this can become dysfunctional in disease. This can be an acquired problem to red cells in many different conditions. And that the red cell, we'll discuss briefly how the antioxidant systems and energetics work in red cells and how they may fail in disease. And lastly, show you information suggesting that how nitrosothiol and nitrosothiol processing in red cells can control energy metabolism and adapt to physiologic stress, specifically to that of hypoxia. And just some fun facts about red cells that people don't think of them, again, as sort of this really highlights the dynamic nature of the erythron, so to speak. It's quite a significant portion of our bodies. There's about 20 to 30 trillion red cells circulating in the average adult. Even though the dry weight is very low, it's about 77% of our body mass and about a quarter of the total cells in our body. We produce 1.4 million red cells a second, 200 billion a day. we replace 1% of the circulating mass a day. So there's actually probably a reason why the red cells turn over so fast. In fact, they need to, in fact, be adaptive. And we produce about 250 kilos of red cells in our lifetime, so a significant amount of energy devoted to this tissue. Lifespan's about 100 to 120 days. Arteriovenous transit's only about 20 seconds, so they're in fact quite quick. And they have roles beyond simple gas transport, as I've mentioned. Regulation of regional vascular tone, a significant role in antioxidant systems. They also participate in self-recognition, and as I mentioned, the physiologic responses to hypoxia, both at the organism, organ system, and regional level. So the principal function, nevertheless, is oxygen transport to tissue. And the principal role of regional vascular tone is to facilitate distribution of that oxygen transport so that it is exquisitely matched both in space and time to need. And this is very well illustrated by work done by Serpil Azurum and Cynthia Beale, where they, although most of us know that flow trumps content in delivery of oxygen to match to need, this is the experiment here was done to illustrate adaptive change in Tibetans to Americans in terms of adaptation to altitude. But the point here is to illustrate the relationship between O2 consumption in forearm and forearm blood flow, which isn't very well matched to hemoglobin concentration, but correlates almost perfectly to forearm blood flow. So really, when we need to improve oxygen delivery to respiring tissue, we dilate a blood vessel and improve flow. We don't, which is also scalable in log orders, whereas the amount of O2 content that can be changed by improving hemoglobin concentration is fairly trivial and isn't really the way we acutely respond. So really what governs blood flow distribution and really the tuning of delivery of blood flow, and perfusion lack must be sensed, flow must be distributed, and really this needs to be effective in space and in time. And in fact, Jonathan here has been the one to, although this was first illustrated by Guyton quite some time ago, the mechanism really wasn't known until Jonathan and David Single illustrated NO processing in red cells that varies as a function of hemoglobin conformation so that NO can either be trapped or dispensed in response to hypoxia, so that red blood cells entering a hypoxic blood vessel leave behind less NO than they encounter, or red blood cells entering a hypoxic blood vessel dispense NO equivalents, leave behind slightly more NO than they encounter when they enter the blood vessel, dilating the blood vessel, recruiting blood flow, flattening the O2 gradient, and then as red blood cells that are well saturated enter a blood vessel, they leave behind slightly less NO than they encountered, so that it acts as a thermostat to exquisitely match perfusion sufficiency to metabolic demand. And this works really has been illustrated just to show you the system that's used, because we're going to use this experimental platform later. These are the way that to illustrate context responsive vasoactivity for red cells. This is a figure from one of Tim McMahon's papers when he was with Dr. Stamler at Duke. An aorta is sliced like a loaf of bread and hung between two hooks. And as there's a constriction that registers as an increase in tension in the vascular ring system, and when there's a dilation, it registers as a reduction in tension. And so as a function of PO2 in the tissue bath, red blood cells are either constrictors, as I mentioned, or dilators. And that this is an endothelial independent response. This is data from Diana Deason, also at Duke, showing that red blood cells will dilate intact rings, likewise denuded rings, while acetylcholine will only dilate an intact ring and not a denuded ring. So that red blood cells are not acting through or causing endothelial NOS to activate, but really dispensing NO that's bioactive from themselves. Really about 1 in 1,000 hemoglobins is carrying these NO groups. It's quite quite potent. The concentration really is about 450 nanomolar in blood for the bioactive form, the S-ligated form, nitrosethiols. Really only about 0.1 to 1% is released during circulatory transit. And one of the first projects that we did exploring this was trying to catch the RDRF, which we might call it, or the NO that's exported from red cells itself. We developed this system where we could sort of imitate circulatory transit for red blood cells. This is a cylinder that spins underwater so that it is temperature controlled. And with a gas blender, we manipulate the gas that's flowing through the center of the chamber. And so by deoxygenating the flushing gas, we deoxygenate the red blood cells themselves, which form a film as the cylinder spins. So over time, the PO2 will fall, and we can recapitulate the deoxyhemoglobin desaturation curve. And so the way we went about this experiment, we put red blood cells in suspension with a bait thiol reactant outside the red blood cells, deoxygenated them, and looked by LCMS to see if we could measure the NO that was caught on the bait. And what we were able to show, in fact, that as we deoxygenated the red cells in suspension, we saw an increase in NO groups on the bait that in fact looks exactly like the deoxyhemoglobin dissociation curve. And over time, as we deoxygenate those red blood cells, we see NO disappearing from inside red blood cells and appearing on the bait outside. So this is the RDRF, as we call it, or the vasodilator that red blood cells are exporting in terms as they deoxygenate. Now, the bioavailability of NO in plasma, as we mentioned, is controlled by red blood cells. In fact, as red cells parse NO groups from either within to without red blood cells, that parsing function can vary in disease. And these are the same data almost, just put a different way. Here we plot the total NO content in the system in the spinning tonometer, really is a function of O2 content in the red cell. As the red cell is fully saturated, all the S-ligated NO is inside the red cell, and as we deoxygenate the red cell, we see it appearing in plasma. Whereas if you take red blood cells from humans with sepsis or septic shock, that parsing function is biased. And in fact, it's biased in a way that's consistent with the physiology that we see, dilated distributive shock. So even when the red cells are fully saturated and they should not be exporting any NO, they in fact do, and in fact they export it more avidly than they do in normal conditions. This is also consistent with the vascular responses that we see. So when we look at the ring preparations, this is the percent dilation from normal red blood cells. This is the percent dilation from red blood cells with septic shock. If we illuminate those cells with UV light and get rid of all the S-nitrosethiols, that response disappears. If we load the red blood cells with with nitrosethiols with a SNO donor, then we recapitulate the response that we see in sepsis, and the opposite occurs in the lung. When we look at hypoxic pulmonary vasoconstriction in an isolated perfused lung bioassay, we see the normal response of vasoconstriction with hypoxia in normal red cells that's blunted with red cells from septic shock, that is amplified when we deplete red cells of NO, and then goes away again as we load the red cells with SNO from an NO donor. So we wanted to look at sort of how this might be happening if we examine membrane thiol status, and I blew through that diagram before, but basically there's the export system is believed to work through membrane trans-nitrosylation reactions from within to without red blood cells, exporting NO groups through a membrane thiol. And if we look here at free thiol status in normal red blood cells and the amount of NO that's on the red cell membrane, it's fairly small. And if we look at the same features in red cells from humans with septic shock, we see depletion of free thiols and an increase in S-nitrosethiols on the membrane. We tried to model this. We took red blood cells and exposed them to superoxide through hypoxanthine system, which generates superoxide really at the levels that we see encountered in oxidative stress. And we're unable, in fact, to reproduce this injury, although we could decorate the membrane with nitrosethiols. And it caused us to wonder, what really is protecting the membrane thiols in red blood cells. So we went to explore the antioxidant system in red cells, which is powered by glycolysis. And I'm going to take a diversion and go through red cell biochemistry briefly. So what we had to do really to injure the red cell membrane and recapitulate the injury that we found in human sepsis was first to deoxygenate the red blood cells. So we put the red cells back in the spinning tonometer. These are the oxygenated red cells, and looking at the density of reduced thiols, you can see they're quite resilient to superoxide, yet when we deoxygenate the red cells, they lose that resilience. And so in order to sort of explain that phenomenon, we need to talk about how glycolysis is regulated in red cells and why glycolysis is so important to antioxidant systems and maintenance of the membrane thiols that are key to nitrosylation-based signaling in red blood cells. So basically, the protein that acts as the anchor for this metabolic regulator in red cells is the band-3 protein, or AE1, the anion exchange protein. It forms this, or is the basis for this complex. It facilitates gas exchange and metabolite conversion in red cells. It optimizes heterotropic effects upon hemoglobin oxygen affinity. It is a tetramer. It is also attached to spectrin and ankyrin, glycophorins, CD47, and RH complex, so it's quite a involved protein complex. There are 3 main domains: a membrane-spanning domain, a C-terminus, which is binding carbonic anhydrase, and an N-terminus. Both of those are in the cytoplasmic domain. They bind this glycolytic complex, which we'll talk about in some detail. And as mentioned, the function really is to facilitate the Bohr effect shown down there, which I'm not going to go into, and also this regulation of glycolysis, which in red cells, thankfully, is fairly simple. So red cells, ironically, are an anaerobic tissue. They don't use oxygen to produce energy. They make ATP only through glycolysis, and it's fairly simple. There are 2 branches, the Ebden-Meyerhof pathway and the pentose shunt. The Ebden-Meyerhof pathway is illustrated here in orange, and this set of enzymes in the terminal limb of the Ebden-Meyerhof pathway are important for its regulation, as I'll show you in a moment. The pentose shunt is an alternative branch at the proximal end of this pathway, really where these 2 branches compete for glucose 6-phosphate as substrate. The pentose shunt, one of the earliest enzymes is glucose 6-phosphate dehydrogenase, and this is the sole means for NADPH recycling in red blood cells. NADPH recycling is essential for thiol-based antioxidant systems in red cell, which are really is the principal form by which red cells deal with superoxide and hydrogen peroxide. So superoxide dismutase and catalase essentially convert superoxide to water, but glutathione reductase is required to keep this system running. And that system runs on NADPH, and NADPH can only be recycled through the pentose shunt. So essentially, the pentose shunt is essential and has to be fairly robust and must be able to turn over NADPH rather avidly in order to deal with oxidant loading. So this is interestingly regulated by hemoglobin conformation in red blood cells. The band-3 protein, the cytoplasmic domain, recruits these terminal enzymes in the Ebden-Meyerhof pathway when this binding site is open, and when red blood cells deoxygenate, Deoxyhemoglobin effectively competes for that binding site with these enzymes, disperses them into the cytoplasm, so that in arteries, really where red cells are encountering higher oxygen tensions, the Epstein-Meyerhof pathway is shut down. The pentose shunt has unfettered access to glucose 6-phosphate, and there's robust ability to recycle NADPH. In veins, the Epstein-Meyerhof pathway is activated because hemoglobin disperses this enzyme set in the terminal end of the Epstein-Meyerhof pathway. This is in fact where NADH is recycled and ATP is produced, and NADH is what powers methemoglobin reductase. So in veins, energy metabolism is devoted to reducing methemoglobin that's generated during circulatory transit. ATP is produced, and in arteries, energy metabolism is devoted to recycling NADPH and dealing with the superoxide load. And this is nicely shown by Phil Lowe's group, where he's showing that in oxygenated red cells, this set of enzymes localizes to membrane, and deoxygenated red cells, they're dispersed to cytoplasm. So red cell energy metabolism oscillates during arteriovenous transit. And in fact, this is relatively important. Now, we look to see how hypoxia, persistence of deoxygenated hemoglobin in arterial blood, might influence resilience to oxidative stress. As you might imagine, if you've still got deoxyhemoglobin around, then the Epstein-Meyerhof pathway never gets shut down, and we've got constraint in NADPH recycling, and that this ought to limit the ability to protect membrane thiols, which is why we were able to only injure the membrane in deoxygenated cells. So if we look at the ratio of reduced to oxidized NADPH and oxidized to reduced glutathione, don't ask me why the convention is to present those in opposite fashion. It's a little confusing, but that's unfortunately the way it's done. In oxygenated red cells, when we confront them with a load, and over time here they're being exposed to superoxide through the xanthine oxidase hypoxanthine system. Oxygenated red blood cells, you can see that they're able to maintain reduced NADPH and reduced glutathione. Yet when we deoxygenate those cells, they're unable to do so. If we pretreat those cells with carbon monoxide to keep hemoglobin off band 3, so if we treat the red blood cells with carbon monoxide, hemoglobin stays in oxyconformation even when we deoxygenate it. The Epstein-Meyerhof pathway enzymes remain complex to band 3. The pentose shunt should stay active, and in fact, deoxygenated red cells are rescued by pretreatment with carbon monoxide. Likewise, if we use the compound DIDS, which binds to the exofascial domain of band 3, altering the conformation of the cytoplasmic domain and preventing hemoglobin from binding, but not preventing the glycolytic complex from binding, we likewise rescue the cells. So basically, deoxyhemoglobin is the way that deoxygenated red cells become vulnerable to oxidative stress. And we can produce the same effect if we block the pentose shunt with DHEA, which is a G6PD inhibitor. In oxygenated red blood cells, if we block them, we make them look like deoxygenated red cells here. And likewise, if we block GAPDH with a selective inhibitor, keningic acid, we can rescue deoxygenated red blood cells and make them look like oxygenated red cells. So the deoxygenated red cells, if we interfere with the Epstein-Meyerhof pathway at a proximal point at GAPDH, we rescue them. In oxygenated red blood cells, if we interfere with the pentose shunt, we make them appear like deoxygenated red cells. And again, the same thing happens. We protect the membrane thiols with the same effect. So we wanted to see if this is disordered in disease, and the model system that we began looking in was sickle cell disease, essentially because the binding affinity for sickle hemoglobin for band 3 is increased, and so this ought to, oxygenated sickle red cell ought to look like a deoxygenated normal red cell. And so we approached this with the following hypotheses: that sickle hemoglobin binds more avidly to band 3 than does normal hemoglobin, and therefore veils the binding site for GAPDH on the membrane. Therefore, the Ebden-Meyerhof pathway should dominate glucose metabolism in sickle red cells regardless of O2 content, and that sickle red cells under oxidative load should not be able to accelerate HMP flux or the pentose shunt, and that thiol-based antioxidant systems in sickle red cells should lack capacity to regenerate oxidized membrane thiols. And go on from there. So first, just to show you again, the ability for sickle red cells to protect membrane thiols is lost. If we look at the redox poise in the membrane under normal conditions, it's not that different. However, the percent that sickle membranes lose when confronted to oxidative loads is significantly more than normal red blood cells, and that this has functional significance. They are more vulnerable to osmotic loading. So these are the lysis curves for normal red cells when confronted with an with an osmotic load after they've been confronted with superoxide. It doesn't really shift much, but a sickle red cell shifts quite a bit. And again, these are the redox potentials in the cells. Here we use something called the half cell because the reducing equivalent concentration is different between the two populations. More negative is good, so it's just like a battery. Reducing power is described in voltage, so the more negative the cell is, the better the more reducing power it is. So here are normal red cells. When we confront them with superoxide, they lose a little bit of reducing potential. You take a sickle red cell, they're at a resting state, they are less negative and less able to maintain their reducing power, both looking through the glutathione half cell or the NADPH half cell. And again, if we block GAPDH in these cells, then we can rescue them. So we use koningic acid. Here are the normal red blood cells when we confront them with superoxide. Here are the sickle red cells when we confront them with superoxide losing reducing power. And if we treat sickle red cells with koningic acid, which interferes with GAPDH function, then they are rescued, and likewise we prevent them from losing membrane thiols. And in the interest of time, I'm just going to go right through the binding studies. Suffice it to say, the sickle hemoglobin veils this GAPDH binding site, and the sickle red cells lose the ability to vary flux through the Ebden-Meyerhof pathway, as mentioned. And this just shows it again, visually. You can see that here's band 3 immunostaining as a function of oxygenation. You can see that that's unchanged. Whereas in normal red blood cell, when we oxygenate the cell, GAPDH is recruited to membrane, but in sickle red blood cells, it remains dispersed. One thing I do want to show you, because this is really the means by which we illustrate nitrosothiol-based control of energy metabolism. We developed a method for stable isotope tracking of glucose through the two glycolytic branches. So we take C13 labeled glucose, which is the second carbon in glucose, and if that glucose is metabolized through the Ebden-Meyerhof pathway or through the pentose shunt, there's an isotopomer of lactate that can be distinguished by NMR so that we can tell if the lactate came through the pentose shunt or through the Ebden-Meyerhof pathway. And in this way, we're able to look at relative flux through the pentose shunt in intact red blood cells. So the first thing to notice as we're looking at sickle red cells and normal red cells is that the resting sort of energy metabolism in sickle red blood cells is very different from that in a normal red cell. Here we plot glucose uptake versus lactate production. And so up and to the right is higher sort of RPM, so to speak, for glycolysis in the red blood cell down and to the left. So these red cells, normal red cells, are really running at lower speed, and suggesting that there's inefficient glycolysis. Here we plot proportional HMP flux. So this is the amount of glucose that's going through the pentose shunt or the hexose monophosphate pathway relative to the amount that's going through the Ebden-Meyerhof pathway. And as I've mentioned before, in the oxygenated red blood cells, if we stimulate the pentose shunt, it can accelerate. Here we use methylene blue to do this. In deoxygenated red cells, the stimulation is much less effective. In sickle red blood cells, even oxygenated red cells cannot accelerate the flux through the pentose shunt. They can't recycle NADPH. And again, the antioxidant system is depowered because glycolysis is impaired. This is also shown, in fact, occurs in stored red cells. And you can see that with storage, in fact, there's a progressive loss of the ability to cycle glucose through the pentose shunt. So this is really what I'd like to sort of was trying to set you up for, is adaptive control of energy metabolism through red cells. And it occurred to us that there are 4 key enzymes or 4 key control points in the system where there is an S-nitrosylation motif. And in fact, GAPDH has been shown to be regulated by S-nitrosylation. So we applied a CSNO or an NO, a nitrosylethyl donor, to red blood cells to see if we could rescue antioxidant systems in deoxygenated red cells. And indeed, just like if we use koningic acid to block GAPDH, if we use CSNO, we can make a deoxygenate, we can restore resilience to oxidative stress in a deoxygenated red cell. And in fact, when we look at GAPDH activity and we add CSNO, we can depower GAPDH, which we can recover by using a reducing agent. And this has been shown, in fact, and published by Jonathan some time ago. And if we look at HMP flux in the same situation and we add CSNO, we can, in fact, accelerate or improve the acceleration of the hexose monophosphate pathway. If we use CSNO, in fact, we can improve it particularly well in deoxygenated red blood cells, and that this is a dose-dependent effect. And we restore the ability to recycle GAPDH at the same, restore the ability to recycle glutathione when we use CSNO. So this is the half cell. You can see it's relatively lost. Reducing power is lost when we confront cells with oxidative loads. And the ability to maintain the half cell is improved when those cells are pre-treated with CSNO. So how does this fit into red cell physiology and what might be the point? And so what I want to suggest is something, this is the controversial point and for which the data is a little thin, but I want you to consider this paradigm and suggest a signaling loop between red blood cells and kidneys that is intended to enable red cell metabolic adaptation to increased O2 transport demands. So superoxide is produced about in 10% or so of the events where hemoglobin conformation switches. So about 10% of the oxygen that comes off oxyhemoglobin is superoxide. The need to deal with superoxide, therefore, is significantly increased under oxidative stress. So really the pentose shunt needs to be relatively unfettered in settings of hypoxia. So here's the evidence suggesting that this might be true. We know that red cell signaling is involved in HIF stability, both from a group in Toronto and also Lisa here, who's shown this in her JCI paper, that EPO itself is a HIF responsive element, that EPO is known to activate eNOS. Red cells, it is believed, may harbor a functional eNOS, at least in a subpopulation. That red cell eNOS, in fact, is membrane associated and may be part of this metabolon that was mentioned. That EPO has been shown to alter glucose uptake in red blood cells. that EPO has been shown to improve glutathione recycling in red blood cells, and that EPO has also been shown to activate eNOS through the PI3 kinase AKT pathway as it does in other tissues. So here are the data suggesting that this might be true in red blood cells as well. So if we take oxygenated red blood cells and we stimulate them with methylene blue, which is the way that we accelerate or pull on the pentose shunt, we take oxygenated red blood cells here, and we either use an arginase inhibitor to improve arginine availability for NOS, or we use, this should say LNEO, or a NOS inhibitor. Really, we don't influence things very much. But in fact, if we, with NOHA, which ought to improve eNOS output, we improve HMP flux in methylene blue stimulated red cells, and we prevent that improvement if the NOHA treated red cells are also treated with LNEO. In fact, if we then use CSNO, as I told you before, we can rescue deoxygenated red blood cells. If we activate eNOS with EPO, we likewise improve HMP flux. If we use wortmannin, which is a PI3 kinase inhibitor, we block the ability of EPO to improve HMP flux in red cells. Likewise, with LNEO, we block that ability. If we take, now, further suggests that this is not an authentic NO effect, but in fact an S-nitrosylation effect. If we take the CSNO-treated red cells and treat them with UV light, we reverse the rescue effect that we showed before. Likewise, if we take EPO-treated red blood cells and illuminate them, we reverse the rescue effect. And likewise, if we disrupt the metabolon with a band 3 agent that not only prevents hemoglobin binding, but also prevents GAPDH rebinding, and use carbon monoxide to prevent hemoglobin-based S-nitrosylation that may be facilitated by hemoglobin in the setting of eNOS activation, we prevent this effect. So together, these data suggest that red blood cells are EPO responsive, that EPO activates eNOS, and in coordination with hemoglobin, S-nitrosylates proteins in red blood cells that unfetters the hexose monophosphate pathway in deoxygenated red blood cells, and therefore adapting or enabling deoxygenated red blood cells to adapt to anticipated oxidative loading in the setting of hypoxia. So I'm hoping that after this sort of deluge of information, you'll continue to consider the following: that there's a very adaptive ability within red blood cells which coordinate physiologic responses to hypoxia, that they do exhibit acquired dysfunction, and that there's an adaptive control in red blood cells which enables energy metabolism to change in the setting of physiologic stress. And in fact, what we haven't gone into is how this really is a function of subpopulations within red blood cells. As I mentioned, there's a very rapid turnover. The population of red cells with EPO responsiveness is probably only about 2% of relatively juvenile circulating red cells, and that this isn't really an entire red cell population response, but a subpopulation. So with that, I'll thank you for your attention, and I'd like to acknowledge all the people that did most of the work. Really, the advice from two people at Washington University that's been invaluable, Allan Schwartz and Bill Frazier. Andre Davignon and Xia Ji did the NMR measurements, and these are all the people that did all the work in the lab, and the people who've really had the good ideas and given me excellent advice over the years. So thanks for your attention. Be happy to take any questions.
Thank you very much, Allan. That was terrific. Everybody in the room probably has about 30 or 40 questions, so we'll have to keep it relatively brief. But I guess by virtue of having the non-functional mic, I can still ask the first set of questions. And well, one of them is just thank you for mentioning Brian. You know, he passed away recently, and he what a great individual.
Absolutely.
A tremendous scientist, and contributed to both of our careers, to a lot of the careers in this room. Let me just give you a slow ball over the plate here and ask you to talk about where red cell eNOS is in relation to the hemoglobin, and how the NO coming off, or any kind of red cell NOS if it's not eNOS, and how NO coming off of NOS would be turned into nitrosonium that could modify a thiol, and ask you whether methemoglobin is involved, and whether there's an AV gradient in methemoglobin or an abnormality in methemoglobin in sickle cell that informs this chemistry. Those two.
Nice slow ball, Ben. So, so those are all great points. So without going into a lot of the details, it appears that methemoglobin uniquely facilitates activation of NO that's produced by NOS to a nitrosonium, and to facilitate or nitrosylation of N, and that hemoglobin, presumably, the NOS which has been localized to membrane, not been localized to this metabolon, so to speak, that there's either full hemoglobin tetramers or alpha beta dimers that are bound to membrane locally with NOS that are really there just to facilitate S-nitrosylation rather than be in relation to oxygenation. And that the conformation changing with band 3, so band 3 itself changes conformation with oxygenation, deoxygenation, and it is attaching and deattaching from the cytoskeleton. And so this metabolon is assembling and disassembling with oxygenation. And presumably, at least the concept is that only as red cells deoxygenate, eNOS would be located proximally with hemoglobin so that it could nitrosylate GAPDH and decelerate the Erdős-Marhof pathway in hypoxia. I don't know if it was evident when I showed you that eNOS really doesn't do much in oxygenated red blood cells to glycolysis. So really, it probably isn't sort of localized in a way to facilitate the nitrosylation event. But none of this has actually been shown.
but methemoglobin. So if iron 3 reacts with NO to enable the iron 2 NO plus complex to transfer, is that part of the equation? You would assume that there'd be more met in arterial blood given your data.
No, well, no, the other way around. So it has to be created in the course of deoxygenation. So as the red cells are emerging through the lungs, all the MET has been converted on the way back from tissue to the lung. So that's when the NADH or methemoglobin reductase is active, and so after coming through the lung in oxygenated red cells, there's virtually no MET. Then MET starts to form as the red cells deoxygenate, and now the eNOS, if activated, can nitrosylate the proteins, whichever ones they are, that are involved in regulating glycolysis. And in sickle blood, okay, none of this can work because the band 3 binding site is capped by denatured hemoglobin. So the system is, prevented. The things that need to come together presumably can't.
I think this works now.
You looked at gender differences in the system, and is there any differences with normal physiology, such as pregnancy, aging, menopause, and stuff like that?
So probably. So one thing we do know is So the paper that we did with Serpil, where we were looking at human adaptation to altitude, there's a gender-based difference in one, snow metabolism in red cells, and two, the EPO response to altitude. So women are presumably better able to do this type of signaling than men, although I haven't looked at, by the way, these are all boy red cells that I showed you. They aren't girl red cells. We haven't done enough of this to really look and see what the gender-based difference is, although there probably is one.
Pregnancy or aging, I mean,
No, we don't have a protocol to get blood from pregnant women. And these are all, healthy volunteers from the lab. So most of us are not aged enough, really, I think, to show the age effect. Yeah.
I just have a quick question.
Sure.
I am a convert to the erythritron. In fact, I think it should be expanded to the erythnome or something like that. So I was wondering, isn't it a little frustrating to know all this stuff about sickle cell and yet know that because it's binding to the band 3, you're sort of stuck at that point? I mean, you don't see a treatment there, do you?
Yeah, right. So what could you do about this? So people have tried a couple things. So it's been known for some time that sickle hemoglobin is complexing with band 3, and that this was originally thought to be a problem with rheology in sickle cells, and that people have tried cyanate initially to change hemoglobin conformation and get it off. It doesn't work very well. Obviously, the drugs that we would use to manipulate GAPDH are not exactly things that we can do, since it's a relatively ubiquitous enzyme and we can't selectively interfere with it in red blood cells. However, there are small molecule SOD mimetics that we're starting to explore as a way to at least dampen the effect of superoxide in sickle red blood cells. And so we don't really have a handle on how to rescue metabolism in sickle red cells, but we might be able to at least, focus on the superoxide.
Thank you.
Yeah. Dennis, yeah.
Here's the mic. This works, so people can hear you back.
Your hypothesis about the CO, the way it's working is strictly through preventing the NO nitrocele formation? Because I wondered if it had other effects on conformation that would, be an alternative explanation for how it's working in the system.
Yeah, good point. So, in fact, one thing that I think I know, but in fact would like to know from you, is whether or not carbon monoxide interferes at all with NOS function, because I couldn't find that it does. My impression is that it doesn't, but I'd be worried a little bit that we're in fact influencing NOS activity with carbon monoxide. So it may be working two ways. One is it locks the hemoglobin into R state. So the transition to T state is really what ought to, enable the cooperation with NOS in terms of making a nitrosothiol. Likewise, by sort of veiling the heme itself, then it can't participate in abstracting the electron from the NO that's produced by NOS and make a nitrosonium. But right now, it's really just a phenomenon that if we use carbon monoxide, we both, quote, prevent the effect of deoxygenation on the EMP, and also we prevent the effect of what appears to be eNOS activation. But we couldn't do it with carbon monoxide alone, what I didn't show. So we use carbon monoxide alone. We don't block the effect, and presumably because GAPDH can rebind to band 3, we had to both alter band 3 conformation and use carbon monoxide to block the EPO effect on glycolysis.
I wondered about that GAPDH binding phenomena. I think that's been seen in other cell types too, where it shifts between membrane and cytosol. Is there any parallels or, common mechanisms, or are the red cell, very unique in how they do that?
Well, my understanding is that band 3 is also ubiquitous, and at least where I think it's been explored is in kidney, and where there's a band 3 GAPDH membrane sequestration, and so I think it's the same mechanism.
Thanks, Alan. That was awesome. I'm confused about the eNOS under hypoxia, though. Are you saying that eNOS is contributing under hypoxia? Because, as I understand it, eNOS activity drops under hypoxia, and it's really substantial at 80% SO2, which would kind of mean that, can you help me understand that a little bit?
I can try. So I don't know. So here's the basis. So the way I see it is a crosstalk between red cells and kidney. So that there is hypoxia or tissue hypoxia, which either can come from anemia in normoxia, okay, which is the easiest one to understand. So PO2 is normal, but tissue O2 delivery is impaired. In that setting, we do know that HIF can be stabilized through PVHL S-nitrosylation in normoxia, presumably through an interaction with red blood cells. This has come from Phil Marsden and Greg Hare in Toronto. And that EPO is then produced. This EPO then signals to red blood cells that in anemia, it's expected that each red cell will desaturate more fully, more superoxide will be created, and the pentose shunt needs to be able to accelerate further. So that as those red cells desaturate, EPO is activated eNOS, which slows GAPDH down, allowing the pentose shunt to run faster. So it's easier for me to understand in normoxic anemia. So that kind of makes sense to me. In hypoxia, okay, if PO2 in arteries is low, as well as there's a lot of desaturated hemoglobin, my assumption, while it's not demonstrated, is that the PO2 is low enough to cause EPO response from the kidney, but not so low in veins, okay, that where the PO2 is quite a bit higher than in tissue, that eNOS is still able to generate NO. Because the PO2 really in veins and hypoxia is still, 30 or 40. It's not, down around 20, 10, where it gets in tissue and eNOS is affected. I don't know, Dennis, you're the man here. What do you think about that?
The funny thing is, out of the 3 synthetases, the eNOS is set up to have the best response at low oxygen. So the oxygen KMO2 for eNOS is almost completely dependent on its affinity, the iron's affinity, the heme iron affinity. And so we've measured that, and it's down at the single micromolar level. So that's quite low compared to the normoxic levels. So I think it would be set up, my guess is the eNOS is set up good to make NO at these lower concentrations. And it probably would make significant NO, in these kinds of hypoxic conditions you just mentioned.
So, if you expose red blood cells to hypoxia, presumably you're going to be losing snow content from the cells, is that right?
Well, it depends. You might, in fact, make it.
Right. So does a
yeah.
NOS inhibitor influence that process then? Look, Alan, the reason I'm asking is I know
okay.
people
Yeah.
who've tried to find enos in red blood cells and just can't.
Right.
You know, they swear it's not there.
Yeah, let me sort of back up. So first of all, I'm not suggesting this enos is creating the NO that's responsible for exported NO that affects hypoxic vasodilation. That's not at all what I'm suggesting. What I'm suggesting is this enos is placed to coordinate glycolysis in red blood cells. It's also been shown to influence spectrin association with band 3 so that it makes the cells a little more fluid. So what I'm suggesting is that enos in red cells regulates red cell ability to, one, deal with hypoxia metabolically, shifting glucose metabolism towards the pentose shunt from the Ebden-Meyerhoff pathway and not creating the NO that's vasoactive. That, I mean, I don't have any reason to suggest that that's occurring.
What is though?
Pardon?
Where is that NO plus on the beta-93 cysteine coming from then?
Which, I'm following you.
The NO on the beta-93 cysteine.
Where's that one coming from?
Where's that one coming from?
Well, presumably that's NO that's been picked up, NO equivalents that have been picked up in the course of circulatory transit, presumably in the lung. So that the lung endothelium's generating that NO. It's picked up as the red cells course through the lung and then being dispensed by red cells as they sort of encounter vascular beds that have perfusion insufficiency. At least that's the model. So that red cells are moving NO from the lung to tissue selectively where vasodilation is needed. But the specific NO groups haven't been tracked, so I don't have proof.
You guys
for
have to
that.
take this argument outside,
Yeah.
I think,
Yeah.
later on. But I think so it's a we could talk all day about that. That's terrific. I think we have time for maybe to hear from one more person, and I want you to comment, since I see Pete Correida's here, about whether you think that the system is dysfunctional in cardiac bypass pump. But do that, and then one other question.
So what would happen during bypass? Well, a couple of things. First of all, and it's, I don't know for sure, it's fairly complicated. One, temperature is going to influence this quite a bit. So a lot of these patients are cool. There's less within each red cell, less cycling going on, meaning each red cell is not desaturating as fully as it is during normal circulation. And really, this is a two-stage engine. The red cell needs to deoxygenate, reoxygenate, and deoxygenate to, in fact, convert NO into a vasoactive element. In addition, the red cells presumably are picking up the NO in the lung, and the lung is bypassed in bypass. So they progressively, as they sort of dispense what they've got, become depleted. And presumably you could fix that by running NO gas or a nitrosating agent through the bypass lung. Lastly, what likely also happens in red cells that in bypass that really interferes with this system is that microvesicles are forming through shear stress, and that these are budding off and really don't have all the machinery for NO processing, and so they're really just circulating NO sinks. So that if there is any NO that's being dispensed by red cells, it can be trapped by the microparticles that are forming in the course of bypass. So there's really a problem with the system in that setting.
We have one more question over here.
So is there any benefits to use CSNO versus GAPDH inhibitors in terms of protection of red blood cells from oxidative stress or restore SH bonds? It's the first part of the question. And second question, you said that you don't know where to go with that oxidative stress and say something about SOD mimetics, but have you ever tried antioxidant vitamins, for example?
Okay, so the first one, can you manipulate? So I don't think trying to manipulate GAP-DH is viable. Because you'd have to selectively target red blood cells. Obviously, we'd wreak havoc with if we, interfere with GAP-DH function in a non-selective fashion. I'm not sure how to target red blood cells themselves. And also, it really does, we can't truly shut down the Ebner-Meyerhoff pathway because that's where ATP is made in red blood cells, and likewise NADPH is recycled. So what we need is oscillating flux in the red blood cells. SOD mimetics, I think, are better than, quote, antioxidant vitamins for a couple of reasons. One is the SOD mimetics that I'm referring to are selective for superoxide, meaning they don't react or quench any other reactive oxygen or nitrogen species. And what's been pretty clear in the antioxidant literature is sort of brute force reduction, so to speak, is just as bad as brute force oxidation. That a lot of the redox signaling systems need a little oxidative tone, and that if we quench oxidation-based signaling with promiscuous use of antioxidants, you just trade one problem for another. So these SOD mimetics, one, are selective for superoxide. Two, have sort of a physiologic, they catalyze conversion of superoxide to hydrogen peroxide, and so that they also don't sort of, they don't exhibit a U-shaped dose response. They will continue to be effective. They can be partitioned into cells or into plasma and so on. So I think that these small molecule non-protein SOD mimetics might be the best way to deal with a problem that I think is specifically due to superoxide excess rather than, in an untargeted fashion, taking care of a lot of reactive oxygen species. So.
Okay.
Right, so how does that get dealt with? So catalase is unaffected. So catalase doesn't, in red cells, second most abundant protein, doesn't require NADPH or NADH, so it doesn't need a reducing equivalent. And it's got this, in fact, its turnover is one of the fastest in the human enzyme family. So catalase we've got catalase in red cells, we've got excess capacity. So the choke point is SOD in red blood cells. And glutathione peroxidase and catalase are there to deal with it. And glutathione peroxidase is killed by pentose shunt deceleration. So I think that the idea is to use that we hopefully ought to have enough capacity in catalase so we can use the SOD mimetics. But we've just begun those experiments. None of this is demonstrated yet.
Very well done, Allan. Thank you very much.